So most models are not robust, and this is well known, and everybody who's working with these equations knows this. Even the robust model of bacterial chemotaxis, Leibler and Barkay and Leibler, has robust exact adaptation, but the output, the steady-state output, does depend on the chemotaxis proteins, as you can see experimentally. So now we're going to biochemical details. Why are there biochemical details? And this is a real good system. We were attracted to it because it has these juicy biochemical details. So the way it works is this. It's like the system I showed. It has 3 extra details that are well known for 20 years, well characterized, but we don't ask why they're there. It's not clear. So the first detail is, so the way it works is this. The receptor kinase, the sensor, binds ATP. That's fine. But instead of taking ATP, like any normal kinase, breaking out, taking the P and gluing it on Y, like a normal kinase, does something else. Takes the ATP, breaks it apart, and glues it to itself. So it's autophosphorylating.
But what is the system exactly?
Thank you. This is the, thank you, I should have said. It's the osmotic pressure response system of E. coli. It's a 2-component system. It's not, the 3 biochemical details I'll tell you about exist in at least 6 other systems in bacterial, this senses osmotic pressure and other things. We don't know exactly the signal. Osmotic pressure is certainly something that affects it. There's one that senses light. In another bacteria, there's one that senses oxygen. So this.
But these are all homeostatic mechanisms?
They're all homeostatic mechanisms. So first thing is, this occurs at the signal-dependent rate, is autophosphorylation. The first detail, autophosphorylation. So you glue the P to yourself. The second detail is phosphotransfer. You take Y, and then you pass on the phosphate. Instead of, well, you pass it on to Y. Phosphotransfer. Now, so far, all the bacterial component systems do this. This is all bacterial. And then this one has, the 6 others have a special feature, which is, instead of a special phosphatase doing this arrow, doesn't exist, special phosphatase, it's the same enzyme, is a bifunctional enzyme. It does both, it does 3 things. It does autophosphorylation, phosphotransfer, and dephosphorylation.
It actually accepts the phosphate group back, or it takes it off?
I don't know. But it's not an energy-dependent mechanism. So it has dephosphorylation.
Takes it off.
I guess it's thermodynamically impossible for it to take it back.
I don't think it's reverse. It's not. I don't think
No,
it's reverse. But dephosphorylation. So this is what's called. This is a bifunctional enzyme. It's a bifunctional enzyme. These things exist several places. It's very interesting to think. It has 2 pockets. One that does phosphorylate, does the adds, and the other one removes. So it's doing 2 different things. The same enzyme is doing 2 opposite things. It's not a reaction in its reverse, like any enzyme catalyzes the reaction in its reverse. It's 2 different reactions that it's doing. It's a bifunctional enzyme. It's going to be very important. Bifunctional enzyme. And the dephosphorylation is ATP dependent. Now this is something weird that Guy found in the materials and methods of the papers. You have to add ATP in order for it to do the dephosphorylation reaction. That's weird because it doesn't use ATP for energy. It uses ATP as a cofactor. So it's an ATP dependent dephosphorylation.
It's not using it as a cofactor. So it's not consuming it anymore.
It's not consuming it. Thank you. It's not consuming it. It
Right.
needs it
Right.
apparently.
Right.
It also needs ADP also works, but ADP is in much lower concentrations, 20-fold lower with ATP. So we have our biochemical features. Now you look at this, you say, let's analyze it, solve it. Right? So this is signal dependent. You add the signal, there's more phosphotransfers, blah, 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 you get more output. You take away the signal, dephosphorylation, no more phosphorylation, output goes down, so it reaches steady state. Now, this model has 7 differential equations with a lot of unknowns. You can solve it analytically, you can solve it completely, mass action kinetics, without any assumptions. But I'll show you that you can actually solve it in the blackboard in one line by looking at it in a little bit different way. Instead of looking at it as a signal system, you look at it as a machine that breaks down ATP. Look at it just as an ATPase, this whole thing. I'm going to put a black box around it and ask how many times the ATP they get broken and how many phosphoryls are thrown back into the water. And those two have to balance at steady state. Every ATP broken, the phosphorus has to go to the water. Yeah.
Does this split a new ATP, or does it transfer the actual phosphate?
This is a phosphate transfer. It's the same phosphate goes from here to here to here. It's the same. That's the thing. It's a conservation. And one phosphate goes. So here's the one equation. So the rate of ATP breaking hydrolysis is just the rate V1 of S times X ATP. Now what does this mean? X takes ATP and glues it to itself. So there's no Y here. Because of the phosphotransfer, there's no Y here. It's just X breaking ATP and gluing it to itself. There's no Y on this side. And this has to equal the rate of PI release, of dephosphorylation release, which is the phosphatase rate, times the same thing, times YP. It's actually the ternary complex XATP-YP, but because of its mass action, it turns out to be equal to XATP times YP times something that has to do with binding rates. Now what happens in this very special equation? You can see immediately that XATP appears on both sides because it's a bifunctional enzyme. And it needs ATP for both sides. So we can cross it out unless it's 0. And the solution is the output is a function of the signal, but does not depend on the amount of X, the amount of Y, and the amount of ATP. It's a very weird thing. The concentration scale here, one second, comes from the constants, which are hardwired into what the proteins look like, and has no concentration dependence.
You're assuming here that the ATP binds in the cycle both the kinase and the phosphatase.
It's the same complex. There's only one binding site for ATP on this molecule. It's the same complex that does. The bifunctional enzyme is X bound to ATP. That's what it looks like from the data.
So it can. So with ATP bound, it can either transfer the phosphate to itself, or it can take the phosphate
From YP. Yeah.
Wow.
It's a very unique enzyme, but bifunctional enzymes aren't that rare. If you have a bifunctional enzyme, you have the potential for this crossing out, and you have the potential for robustness. That's what I want to tell you. If you see a bifunctional enzyme anywhere. Each of these three biochemical details is essential. You need all three in order to get perfect robustness. I'm just going to draw, perfect robustness. What do I mean? Instead of this situation, perfect robustness means, I'm going to draw here the rate of the kinase, the signal. No matter what X, Y, and ATP is, the proteins in the different cells, it's the same input-output relationship. Same. If you add additional reactions, like a loss of phosphate here or something like that, if they're small, you approach this robustness. So you can make it as robust as you want by adding additional reactions, but if you add additional reactions, it won't be perfectly robust anymore. It'll be as robust as you make those reactions small. Each of these three details is essential for this feature. For example, if X didn't autophosphorylate itself and pass it to Y, there'd be a Y here. And we won't be able to get rid of Y. There will be a non-phosphorylated Y here. So autophosphorylation does this. If it didn't bind, if this was the form that wasn't ATP bound, and this was ATP bound, we couldn't cross it out. So each one of these reactions, meaningless by itself, makes sense when you look at all three of them together. And I have to say that a beautiful experiment of Eric Batchelor was a robustness experiment, overexpressing X or underexpressing, overexpressing Y and underexpressing, and looking at a correlate of this output, which is the ratio of two genes with fluorescent proteins. And there's a wide range of robustness, experimentally.
So the argument holds it, no matter how many intermediate reactions you put in the system, right?
Good.
So then basically you're going
Wait, let me just explain what you said. You said, this model can be generalized to a whole family of models. If there was a phosphotransfer loop of another W, 20 phosphotransfers, as long as the phosphate doesn't leak out, you can do the black box, and it's the same formula.